Hello everyone, I'm here to go through the exam with you. You should be used to how this works by now. I'm going to do annotations in a blue pen and I'm going to write in the correct answers in a green pen. So let's have a look at question 1 and let's have a look at this diagram. It says it shows how cells from human embryos can be used to grow replacement body parts for humans. So let's just work our way down the diagram. Here you can see a fertilized egg. Now that is obviously a product of the sperm and ovum fusing together, and a more technical term for that is a zygote. Now from one cell you can see the 5-day-old embryo has multiple cells now. So clearly they have increased in number and they do that through division. As we get down to here, you can see some of the inner cells of the embryo are removed, and those are what we call stem cells, embryonic stem cells. Obviously called embryonic stem cells because they come from an embryo. Now you should be familiar with the fact that stem cells are able to differentiate into any type of specialized cell. So the term differentiate means to, I'll just write that down for you, differentiate means to specialize, and we can see some specialized cells down here. Blood cells are specialized cells, nerve cells are, muscle cells are as well. Muscle cells are just down there. Okay, so the first question is about the fertilized human egg. It wants to know how many chromosomes are in there. So the sperm is going to have 23 chromosomes. The ovum is going to have 23 chromosomes. When they combine together to make a zygote, they will obviously have 46. So that's the correct answer there.
Right, he wants to know about the process now labelled G. So let's just have a quick look back, G is this process here. Now, in lessons we learnt about the cell cycle. So really this is a 3-step process. We could call that the cell cycle. Now, step 2 of the cell cycle is mitosis, and that's where the cell actually divides to make 2 more cells, and that's the closest description of what's happening there. And so mitosis is an option, and we'll give that a tick. A 3-day-old embryo. Okay, so the way I worked this out was I just made a couple of notes to the side of the question. I just said, okay, so we started off with 1 cell, then it divides to make 2, then 2 are able to divide to make 4, and then 4 are able to divide to make 8, so it doubles each time. So we've got 1 division happening there, the 2nd division happening there, the 3rd division happening there. So the answer would be 3 because it's divided 3 times. It asks for how many times. Right, it wants to know about the process labelled H now. Let's have a quick look back at the diagram. There you can see that we've already labelled this is differentiation. It's when cells differentiate or specialise into, it's when stem cells specialise into specialised cells. Okay, so we've got differentiation down there, lovely. Which 2 parts will be found in all cells in the diagram? Okay, so let's go through them one by one. Cell membrane, we know that's in every kind of eukaryotic cell. It's in animal cells, in plant cells, fungi cells and protist cells. Cell wall, now you should recognise that these are only found in plants. Same with chloroplasts, because plants obviously need to carry out photosynthesis. Plasmids are actually found in prokaryotic cells. Prokaryotic cells are the kinds of cells that make bacteria, and that means the 2 we're left with here. Hold on, let's just go back. So it wants to know which ones are found in all cells in the diagram. So
these cells, blood cells, nerve cells, muscle cells, these are all animal cells. So you know you're looking for things that are in an animal cell. Right, that's in plants, that's in plants, that's in bacteria, cytoplasm and cell membrane are in animal cells. Okay, stem cells from human embryos might be more useful than stem cells from adults. Well, this is just something that you should know. It's the most widely recognised difference between embryonic stem cells, so from human embryos, and adult stem cells. You would say that embryonic stem cells can differentiate into any type of cell. That implies that adult stem cells can't. Adult stem cells can only differentiate into some kinds. So embryonic stem cells are more useful than adult stem cells if you're looking to treat a disease like paralysis or diabetes or something like that. Okay, some parents have stem cells from the umbilical cord. That is part of the tubing which leads from the baby to the mother and delivers nutrients and water and stuff like that. These stem cells can be used to treat diseases in that child later in life. So the child is born, the umbilical cord and everything is obviously removed, but instead of just putting that in medical waste, you could instead store that and freeze it and then save some of the stem cells for if the child is ill later on and potentially use them. It wants another reason why stem cells from the child's own umbilical cord could be used rather than from another embryo. So let's go through them. Less risk of rejection from umbilical cord stem cells. Stem cells from another embryo can treat more diseases. That's not true. Umbilical cord stem cells are older. It doesn't make any sense. This first one is correct. You need to have stem cells that are a match for your body for them to work. That's the idea behind therapeutic cloning as well. So it's this first one. Okay, moving on. Some scientists have concerns about the use of stem cells. So I think a nice answer to use here would be one that we mentioned before. You could say that they may be rejected. You could say they may not work or have complications. Alright, you could also say is very expensive.
Okay, so this is more of a scientific perspective. Here it wants you to talk about why people object because of religious beliefs, which is more of an ethical thing, not necessarily based in science though. Sometimes they do overlap. It wants an ethical concern about the use of embryonic stem cells. So the idea here is that some people consider stem cells, or I'm just going to put in there embryonic stem cells, to be equivalent to a life, and you could just say therefore using them equals murder, which is obviously very serious. A lot of religious people believe that you shouldn't interfere with contraception, and you shouldn't use contraception. You shouldn't
interfere with conception, which is actually creating a child through sexual intercourse. Alright, moving on to question 2. So this should be pretty straightforward. This is the nucleus, and then this would be a cell membrane. Turn it over. So many types of cell can divide to form new cells. Some cells in human skin can divide to make new skin cells. There are 3 reasons human skin cells need to divide. You need to identify them. So here there are 3 reasons why we need new cells. The best way to remember this is using the mnemonic grr. So you can go grr like that. It helps you remember it. And what it stands for is growth. Let me just switch to my green. Repair and replacement. So you would need new skin cells as you grow. You would need to repair skin cells as they may become damaged. That happens on a daily basis, and to replace them as well, because cells only last for so long and you need to have new ones. That's why you get dead skin forming over your old skin, and then new skin cells are being formed underneath. Alright, so this is an easy question. 2C part 1. Human stem cells may come from embryos. Obviously hair and nerve cells doesn't make any sense there. Human stem cells can be used to treat paralysis, so you need to know that one. And I'm also going to add here and potentially diabetes. There's a lot of information.
You know, it often is with these questions that if you spend a decent amount of time reading through it and looking at the information, then you've probably got a decent chance of getting a lot of marks on it. If you just kind of skip through and skim read it and don't really think about it, then you know, you're not going to have much luck. Alright, so if you want to just pause the video now and just read through it, I think that would be a lot more beneficial than me just reading through the whole thing for you. Alright, so you can just give that a pause and come back to me, and I'll move on and I'll start going through the answers for you. Okay, 3 advantages that you should have seen there of using stem cells from embryos rather than adult bone marrow is that, again, we've mentioned this before, embryonic stem cells can differentiate into any other cell. You can see that's in the text here, right? So that's that bit. Next one, it's quicker, so it's a quicker process. See, that's in the text here, because each stem cell divides every 30 minutes. Here it's every 4 hours. Another one is that they're not going to be rejected. Not rejected as much. You can see that that's in the text. There is a low chance of the patient's immune system rejecting the cells, whereas with adult bone marrow cells, it's a high chance of them rejecting the cells. Okay. Advantages of using adult stem cells or stem cells from bone marrow instead of from embryos. Because remember, adult stem cells, so adult stem cells come from bone marrow. Okay, advantages we've got there is it's cheaper. It's £1,000 for the adult bone marrow, it's £5,000 for the human embryos, or the embryonic stem cells. You can give permission, so
just to expand on that, adults give permission for their own bone marrow to be collected, whereas embryonic stem cells, you know, that's an ethical issue. People think that that might be equivalent to murder. And you could also say that using adult stem cells is safer. It says at the bottom about the safety there. Alright, nearly there, it won't take much longer. So, as an organism develops, here you need to talk about the difference in the organelles. So, specialised cells differ in shape, size, and number and type of organelles. Alright, so if you look at some of the specialised cells below here, they're different shapes, different sizes, and they've got different numbers of organelles. For example, red blood cells here don't have a nucleus, whereas all the other ones do. This palisade mesophyll cell is going to have lots of chloroplasts because its role is to carry out photosynthesis. Alright, so what happens during differentiation to form a specialised cell? You could say that the organelles required, they develop. You could also say the shape and or size changes. So either one of them is fine. Okay, we can quickly go through 4b. A lot of people, for some reason, thought this was a leaf. This is not a leaf. Look at the question. It says it shows some specialised cells. This is not a leaf. Right, this is a muscle cell. These lines going through it, right, they are proteins which allow the muscle to move and carry out its function. Alright, so B is a muscle cell. Its function is contraction for movement. C is a red blood cell. I hope everyone recognised that. And I hope you get the function as well, which is to carry oxygen around the body. This is a nerve cell. So actually nerve cells are very, very long. You can't really see that in the picture, but they go on for a long way. This is actually a very long tail in reality. Obviously it's just a cell, so it's not really, really long, but relatively for a cell it's very long. And they carry electrical impulses around the body. Okay, so remember to go through and correct all of that, and there you go. All right.